Hi all, welcome back to Microbiology Lectures. Last time out we talked about multinuclear or lobe nuclear phagocytes. Now we're going to talk about the mononuclear phagocytes. Typically we consider these the phagocytes that are directly associated with the adaptive immune response. However, realize that even the multinucleated phagocytes, like your basophils, your eosinophils, your neutrophils, they present material to the adaptive immune system also. And so you can get it from many directions. So the mononuclear phagocyte system is the MPS system. Normally we're talking about dendritic cells. Their job is to phagocytize and present material. And so sometimes the multinucleated or lobe nucleated innate immune system cells like basophils, neutrophils, etc., they will just break material down. They'll just destroy, they'll seek, destroy, and they won't present antigens. But the dendritic cells, their purpose is to consume and present. Okay? And so they're circulating in the bloodstream, consuming, presenting antigens. So then the T cells and B cells can then generate antibodies, or so they have a search image, that they can go out and seek the material that matches that antigen. So you have dendritic cells, which really function as these scouts that go out, engulf material, crush it, destroy it via digestive enzymes, and then present it to the adaptive immune system. The adaptive immune system, normally we call those lymphocytes. Okay? And so sometimes dendritic cells are considered lymphocytes. Sometimes they're considered granulocytes, even though they have no granule. So sometimes they're part of the myeloid grouping, or sometimes they're part of the lymph node grouping. And that's species dependent also. Normally when we're talking about this, we're referring to humans, and they belong to the MPS in humans. So that lymphocyte situation is the dendritic cell scouts it out, foreign material, engulfs it, digests it, presents it. The lymphocyte then reacts to it. And there's lots of ways at which reactions can occur. Sometimes that's producing antibodies. Sometimes that is helping other cells by basically marking the foreign material. There's all kinds of things. So, and I also want you to make sure that you understand that not all of the adaptive immune system is not part of the innate immune system. Okay, so we have what we call innate lymphoid cells, or ILCs. Okay, and they are part of that innate immune system. Their purpose is to seek and destroy, and we call those neutral killer cells, or NK cells, and they destroy things that are foreign. And normally they're not specific, they're more generalists, and so they just seek out things that have foreign proteins on the outside of their membranes, and they destroy things that are foreign to them. And so they're really important also in reaction to cancer. So cancer cells, when cells have uncontrolled cell growth, a lot of times there will be unique proteins or unique carbohydrates that are being presented on the outside of these cells, because the cell keeps going through uncontrolled cell growth, and the NK cells can pick up on that, and they will destroy cancerous cells. So it's not just cells that are not self-made, but also they can destroy cells that are self-made or part of your natural system. Okay. So again, back to what we were talking about before. You have an innate immune system, you have an adaptive immune system, and if those two can talk to each other or communicate with each other, you can build up an immune response, and it's a coordinated immune response. So something is consumed, something presents, and now you have memory of that once infection, and so if that infection happens again, the innate immune system doesn't have to consume it again. Just the adaptive immune system will run out and destroy it, and it's a much quicker response. So to do that, we have to have some kind of receptors or some kind of way to present the material. All right? And so a lot of phagocytic cells, like your eosinophils and your basophils and your neutrophils and your dendritic cells, they have surface receptors, and the receptors are transmembrane, typically transmembrane receptors, so they go all the way through the membrane. And when they phagocytize something and they break it up, and then they can present it, they can push material out those proteins and hold them on the outside of the cell. And in doing so, that holding them on the outside of the cell can allow for things to come by and bind or pick up that information. Okay? There are other situations where not just holding the material on the outside of the cell, but producing cytokines, which is a way to broadcast it through solution, either through the blood or through other bodily fluids, and so then you don't have to have contact to contact, but it's instead their cytokines are kind of like a voice, like shouting it out, and things that are around will pick up those cytokines and then act appropriately, whether that flood the region or whether that avoid the region, maybe produce certain type of endotoxin, all kinds of things that can happen. Okay? And cytokines, I don't want to say that they're just part of this kind of immune system. You have cytokines that are triggered anytime a cell really dies. So if a cell dies, cytokines are triggered to notify the cells next to it that this cell is damaged, it's dying, it's dead, and they need to start replicating. So they need to start going through mitosis and replicating so they can increase the number of cells in that region, or heal a wound, or whatever it might be. And then there's a group of molecules that allow for things to stick to each other, adhesion molecules. And these are often useful for things like neutrophils to move from the blood through like the capillaries or through veins out into tissue. And so these adhesion molecules allow for material like white blood cells to bind to epithelial cells, and so that binding allows for them to fit through smaller gaps in some of the capillary beds and things like that. Okay, so let's talk about these pattern recognition receptors, or you remember we were talking about those transmembrane proteins that allow for things to be presented to the outside of the cell's membrane. So if a cell engulfs something, breaks it up, digests it, can present things, okay, this is what we're talking about, the PRRs. Right? They come in really three main types. We call them MAMPs. And so MAMPs are microbe-associated molecular patterns. Right? And so normally this is some component of the microbe that's been digested and then can be presented. Like I was saying before, the most useful component is protein based. That's not always the case. Like sometimes we're talking about other structures, but a lot of the useful MAMPs are protein based. So whether that be lipoproteins or segments of the flagellum, something that's based on protein it is by far the most useful when it comes to inducing a adaptive immune response. Okay. The other two are PAMPs and DAMPs. PAMPs are typically related to pathogens. So they're associated with pathogens, but it's not always exclusive that they're pathogens. But normally that's where the P comes from, is pathogen associated molecular patterns. So meaning that these are disease causing organisms, and your immune system or the organism's immune system has digested one of these disease causing organisms, broke it up, and is now presenting this material. DAMPs, on the other hand, DAMPs are normally released when there is cell damage. So it's like if you cut yourself and the cell is destroyed and it's self-made material typically that is being presented, right? Or other times it might be a combination. Maybe you have an insect that's chewing on a leaf, and some saliva from that insect might mixed with some of the plant tissue, and that can give you kind of a different DAMP. But it's really to indicate that there's cell damage, that cells are dying, and that means you have some kind of response. So a couple things, like when a MAMP is detected, it doesn't always result in immune response. Okay? Sometimes it's just a cell signal to let things know that, hey, there's this component of lipoprotein, and it's not enough to excite an immune response. But when we're talking about PAMPs and DAMPs, almost always there is going to be some type of immune response when it comes to those situations, because it's definitely showing that there is either damage, direct damage, or that this is a pathogen-related organism, and it needs to be basically taken care of. Like, you need to take it serious. The MAMPs, not necessarily, because this could be like native E. coli dying in your intestinal tract, or it could be, some other type of bacteria, that's native to your body, that's sending this signal, and it doesn't necessarily mean that you're going to have an immune response. All right, cytokines. Cytokines are these signal molecules that tell the cells and the surrounding material that something needs to happen. Okay, so you have a few ways at which this works. So chemokines are part of what we call chemotaxis, and that is normally after damage is done. Say you get a sliver, or you cut your finger or something like that. The first thing is to control blood flow to the region by cutting blood flow off. And then the next thing is vasodilation and send blood flow to the region. That's a chemotaxis response. And so with that increase in blood flow, you'll have increase in immune cells. And those immune cells will go to the region and consume, phagocytize, or you may have adaptive immune cells. You will have adaptive immune cells go with, and they may be carrying antibodies that are going to destroy the foreign material anyways. Okay. Now, sometimes what happens is not just a chemotaxis of the immune cells, but sometimes we have a CSF form, and that's colony stimulating factor. So this is what we were talking about earlier, where maybe there's enough damage that you need to multiply certain cells, and so maybe that's a multiplication of cells that would fix the material, or maybe thrombocytes that need to, platelets that need to form and stop the bleeding, or maybe it's a multiplication and differentiation of the leukocytes, the cells that need to either become T cells or B cells. Okay. Interferons, we kind of talked about these a little bit before. Okay, these are things that are going to control viral infections, and so they're reacting to the protein coded, the capsid of, typically the capsid of viruses. Interleukinins, okay, these are produced by the leukocytes, and they're important for transmitting signals between the innate and the adaptive immune system. And then we have tumor necrosis factors, or TNFs, right? And often when these are triggered, it triggers the cell to go through apoptosis. And so it might increase inflammation, it might increase the amount of fluid rushing to the region, right? Along with that, histamines and other things that might make it to the region. But often it will end up being that the cells in that region are either damaged enough, the DNA is damaged enough, or the material is damaged enough that maybe they're bypassing the control system of the cell cycle, and so they need to go through apoptosis. That cell needs to be destroyed. Okay? And so all of these really play this important role. So these transmembrane, so these are showing, this here is showing the TNFs, or the tumor necrosis factors, that move or go all the way through the membrane of these cells. They're interacting with other cell membranes, and depending on the interaction, what TNFR they're interacting with will depend what kind of signal is being sent. Okay? The signal that's being sent to go through apoptosis or necroptos, okay, or signaling the cell to go through proliferation and start dividing. So all these things can occur through these cytokines of different types.